So it's a pleasure to be able to present today. This is a paper that was published recently. I'm an ethicist, so I work a lot on these ethical questions to do with uses of pathogen genomics in general. And in this paper, we're focused on clinical metagenomics. So that kind of setting. We focus on a few different kinds of infections in the paper, but I'm focusing on one case study that we present, which is about a suspected drug-resistant infection. So Ruby, next slide, please. So imagine that you have a case where a patient has arrived at the emergency department and they have a fever, they have respiratory symptoms and some early indications of sepsis. So a sputum sample is taken and it's sent for culture and metagenomic analysis. But in the meantime, the patient is started on first-line broad-spectrum antibiotics. And before the sputum culture itself and the drug sensitivity testing are completed and the results come back, the metagenomics results come back, and they identify Klebsiella pneumoniae with a bacterial genotype associated with extended-spectrum beta-lactamase production. So what do the clinicians do? Well, they increase their infection prevention and control measures. They cease treatments likely to be ineffective for ESBL infections, and that means switching to carbapenem therapy. The metagenomics results are also positive for T. whipple, but that appears to be unrelated to the current clinical presentation. So this case is based on a report that came out recently, last year, Charalumpus et al., and they were looking at a few different cases where metagenomics have been used in this clinical hospital setting, particularly for, I think, respiratory indications. Next slide. And what we're trying to bring out here is really questions about what should the clinician do, what should the microbiologist do, and what are other considerations that might determine when and how metagenomics should be used in this kind of case. So I'll give a little background to start off with. I'll get a little bit into clinical metagenomics and a little bit into bioethics, and then I'll talk about some specific ethical considerations that we should get into. Next slide. So clinical metagenomics involves using, well, it's pathogen agnostic sequencing method, and some of the kind of expected marginal benefits in a clinical setting are the potential for faster turnaround times than traditional culturing or even some other molecular testing methods, identification of resistance genes, regardless, without targeted testing, and addressing diseases of unknown etiology or unexplainable outbreaks. Next slide, please. So I'm not going to get into that too much. I'm not a scientist, I'm an ethicist. What I'm concerned about is the should questions here. What ethicists do in these settings is clarifying value trade-offs. So what different kinds of values are at stake, and what might you be kind of gaining as your benefits if you opt for one course of action, and trading off by not opting for the other course of action. We explore unintended consequences, and we try to provide action guidance to clinicians, researchers, policymakers, and the general public. It's not necessarily about answering right or wrong or yes or no answers. It's about giving kind of generalized guidance and making sure that people are clear on what values are at stake in any given question. I'm kind of iffy about this graphic at the bottom. It's kind of a matter of weighing different considerations. Sometimes that will involve weighing something like clinical effective against health equity, or it might involve kind of the science versus society element, but that's not necessarily always the case by any means. Next slide. So some of the ethical considerations that are raised by that case, we have this patient come in, suspected pneumonia. Well, the metagenomics results have come back and said Klebsiella pneumonia infection, ESBL. The clinicians have already had to do a bit of thinking about a few ethical considerations. Some of those are rationing and cost effectiveness. Some level of resource rationing is required in all the healthcare settings because they're all limited in some way. So clinicians need to decide when are going to be the cases when there may be limited access to metagenomics, or limited number of tests that they can send out per day should be used for which particular kinds of patients under what circumstances. They've also had to think a bit about whether or not this more costly technique produces better results than culturing and resistance testing. That might depend on how time sensitive the case is. It might depend on other aspects of clinical presentation, whether they have a hunch or whether they're really not sure what's going on. And they may need to think about whether the treatment might be rendered much more cost-effective if metagenomics leads to a change in treatment. In this case, we were switching from a first-line broad-spectrum antibiotic to carbapenem therapy. And in that case, we need to think as well about what are the kinds of gains in cost-effectiveness or losses in cost-effectiveness that could be gained through the use of metagenomics and resulting shift in treatment strategy. Next slide, please. A second set of ethical considerations that relate a lot to this link into the question of diagnostic benefit and tailoring treatment. So to what extent does metagenomics give the clinician an advantage in this setting? How much does reduced turnaround time matter? Can it help to avoid ineffective treatment, to reduce the patient's exposure to adverse effects associated with some broad-spectrum antibiotics? And does it prevent their conditioning worsening in that case, or escalation of the case to a more clinically serious one? So these are the kinds of considerations that are raised initially by that case and that the clinician would already have had to think about. There are a few more as well. Next slide. So what was raised at the end of that case was this finding, which wasn't in the original report of T. Whipley. Now, incidental findings are something that's been explored a lot in the human genetics and genomics literature, but it hasn't really been thought about in the context of pathogen genomics, at least in the ethics space. So incidental findings might occur in two different kinds of situations here. The first might be when clinicians weren't searching for an organism that they do discover, or the second when they were searching for that organism, but they're not sure as to how much it relates or is relevant to the current clinical presentation. So T. Whipley is a very good case example of this. A lot of people do carry it asymptomatically, but it can result in Whipple's disease with very severe consequences for the patient. So there's a lot of clinical judgment that's needed here in association with incidental findings. And whenever we're needing more clinical judgment and maybe judgment by the microbiologist as well about how much information to disclose to the clinician, not just the clinician to the patient, we are raising questions about, well, what is the burden that's associated with that additional decision making? Is there an informational burden for the patient if they're told about these incidental findings? And how should we navigate this kind of new information, but possibly new obligations that the information raises as well? Would it be a waste of resources to treat that or not? So those are ethical considerations that might arise in the moment when the results come back. And finally, next slide. We can think about addressing AMR more broadly. I don't want to get into the public health ethics considerations too much in this presentation, but suffice to say that targeted genomic testing offers higher certainty of the association between resistance gene and phenotype, as far as I gather from my limited understanding of the area. But with metagenomics, since what we're doing isn't a targeted sequence and we're not getting a whole genome sequence necessarily with that gene in there for the particular K. pneumoniae, what we may be finding is a resistance gene that's actually associated with, something else in the sample. So because of that additional uncertainty, there are a lot more questions raised about what should be done and how to respond to that finding. Those kinds of questions might not be raised when we're thinking about other methods that could be used. So we need to think about what this means. Perhaps first-line treatment may have been clinically effective for this patient, and maybe the risks of AMR emergence and spread as a result could be lessened, and they might have been not as high as the metagenomics initially indicated. So at this stage, at least as far as I understand, confirmation is always needed. That sputum sample is always going to be sent off for culture and resistance testing as well, which might reduce the marginal benefit and the cost effectiveness of metagenomic testing over other conventional methods. All of these kinds of considerations need to be taken into account because they affect the ethical weighing and weights that we should give to this kind of a development. And we need to be thinking about these, particularly when, on the other hand, we need to be thinking about hospital-acquired infections and additional uses of metagenomics that might be occurring in that hospital setting, not only for the benefit of the patient, but for the hospital more broadly. As I said, I won't get into that, but it's another area that I'm on a team that's looking at that at the moment. Next slide. So I haven't given you any answers, which is very typical for an ethicist, but these are all the kinds of questions that still need to be addressed. And as I said, work is like only just beginning. This is an agenda setting paper, and it's really outlining a few areas where we need more ethics work done to try to answer these questions. So we're just raising the questions for now, but they relate particularly to clinicians and microbiologists' ethical obligations, public health and AMR, and burdens and benefits for the patients, considering the role of metagenomic testing. Next slide. I want to thank my collaborators on the paper and the funders involved as well. You can go to the Journal of Medical Microbiology to read the full paper. Thank you.